Once again, thank you all for joining us. We will go ahead and begin this webinar on mold and the indoor environment, understanding the risk, presented by myself, Rony, the Quality Assurance Manager of Environmental Diagnostics Laboratory, and Dr. Rajiv Sahay, the Laboratory Director. This is a special webinar because we are actually running it from our laboratory today, so you may hear some beeps and whirs from our machinery and equipment. But I hope it's not too distracting from the information we will be presenting in this webinar for you today. One other thing is that we will also have some questions throughout this webinar, hopefully to kind of gauge everyone's involvement and see that we are properly educating everybody on this important topic. So to begin, these are the objectives that we'll be going over today. In regards to mold and the indoor environment. And you can see that we'll try to go over everything to build a solid foundation in understanding mold and its importance. So Dr. Sahay, I believe the first thing we should go over is, what is mold? I'll let you go ahead and take it from here.
Thank you and good afternoon from the Eastern Coast. This is Dr. Sahay. Well, the very first thing, this webinar is focused on mold. So I deem necessary that it is important to understand what mold is. There are a lot of misnomer out there that whether the mold is an organism or symptom or what it is. So to just clarify it, that mold can be referred as neither like a organism nor a symptom, but what it refers to a growth that is in a thread-like structure typically observed on a substratum in various shape, size, color due to the growth of the filamentous fungi. So filamentous fungi is the organism responsible for growing the mold. So that is precisely we can say that is the definition of mold. In the next slide, I would like to give an important information regarding filamentous fungi because this is the word which I have used to focus the mold. So the filamentous fungi is basically a group of saprophytic or parasitic organism that lives by decomposing and absorbing the organic material in which they grow. So that is a group of fungi which by character or by nature produce filamentous structure, because there are several other fungi, they do not have a true filament and they may not involve in the whole mold causation. So the only filamentous fungi that can cause mold.
All right, so now we've talked about the first part of our title, mold. Let's move on to the next one. Can you explain the indoor environment for us?
And this is also very important to just introduce this topic, the indoor environment. Many a time people ask me that what is indoor environment? What we understand when the word indoor environment come, which is, I guess, buzzword nowadays in media. You see this word quite often. So let me give a precise definition to the indoor environment. Actually, the indoor environment is an overall condition within and around a closed or built environment and its relationship with its inhabitants. That's in nutshell is the indoor environment. Now, in my next slide, I will tell you that the indoor environment and indoor air quality or indoor environmental quality, that is the another two buzzword. So the indoor environmental quality is a term which refer basically the suitability of the occupiable space or occupied space as it relates to the comfort and safety of the occupants. So that is a precise definition of indoor environment quality, that is also referred as IEQ. Now, the other sister term is indoor air quality. So think about like a bigger set of picture and then narrow it down to a very specific set of picture, such as the air. So in environment there are a lot of other component, but when it boil down or just pointed to the air and then quality of air, so then it is referred as to explain overall condition of air within and around closed structure with a special reference to health and hygiene. That should be a precise definition for the indoor air quality. You understand that, Ronnie?
Yeah, that makes perfect sense. And now, before we go on to our next topic, we're going to go ahead and do that first question I told you about at the beginning. So I have it up. What is mold? Go ahead and pick the correct answer, and we'll see how everybody does. I'll go ahead and give everyone about 30 seconds. Okay, looks like most everybody voted, so I'm going to go ahead and close the poll. And let's see how everybody did. We had 93% of you getting it correct. It is a fungi. So now we will go back and continue our webinar. So, Dr. Sahay, can you talk about the history of indoor air quality?
Well, the history of indoor air quality as a whole is not that new, which people think. It is known for, like, time immemorial, I would say. And then if we go through the references, there is rhetoric of references available when you can think about indoor air quality. I have picked one reference which goes dead back to, 1500 before Christ. In Egypt, the people were suffering from the respiratory ailment that might be the reason for, various mason and miners work, which also implicated in the Rome or Roman era, that people were also feeling respiratory illnesses. And then the also, in Lucretius has focused this particular topic before Christ 55. So this is quite old. In modern time, basically what happened, people were just decline of the good practices, and the health and hygiene practices were decline, and that may become a major reason for the deteriorating quality of environment and air as well. But in modern age, actually there was a limited scientific approach implemented to understand the fact and differentiate between friction and fact. But in the modern age, as we are getting more educated, then the most of the exploration start happening on the basis of science or scientific principle. And in recent times, when we design the energy conservation in the building and the health and hygiene related issue, then we start finding that actually 1 in 3 building classified as being sick according to the World Health Organization. So that was a great finding. But in the recent time, we start, as I said, linking it with the scientific approach. So many a time it is evidence based. Well, at this point, I want to clarify that the common contaminant for the indoor environment is two types. If we go the origin of the contaminant, the one group of contaminants are having a biological origin, meaning that there is no life in their origin as such. And then there is a whole another group which is biogenic in origin, meaning they must have a biological origin of source. For an example, in the abiogenic, you can possibly think of various kind of environmental factor, too hot, too cold, various kind of chemical. These can be cited good example for the abiological contaminant. Whereas the biological contaminant, as I said, that there must be a life in that, or must have a origin, must be originated with source of biological entities, that is maybe viruses, bacteria, pollen, mold, and insect biodetritus. You can name it. There are several out there that are good example for that. What do you think, Ronnie?
I agree. Where can mold be found in an indoor environment? Or what kind of materials, I should say?
That's a very good and intelligent question, that first people start wondering that where I'm going to meet with mold. Well, you're not going to meet the mold in the downtown, but you are going to meet the mold in around you, maybe in the air, maybe in the surface, maybe in the fluid. So all the three metrics of the environment, you perhaps if there is a mold proliferation, then you are going to meet it because mold is ubiquitous in nature.
Okay. So now we have a little bit better understanding of mold, but there's still some more things we can talk about for it, such as what kind of problems can it lead to when you have mold in an indoor environment?
To answer that question, Ronnie, you have to focus about the catalyst of the issue, like why we are worried about mold and in what case the mold is going to be like going to grow in an environment. To answer that question, it is some very focus or very focal point of that question is that the modern time the buildings start becoming energy tight because we are trying to save our dime and dollar with the energy. So conservation of energy is a good thing. I'm a big supporter of that, but at the same time that if it is being not properly managed, then you have to pay the consequences. The other thing is that they for maintenance, because if your belts are tight, very first thing people will try to take off or check off on their things to do list is the maintenance of building, because that comes last in the priority if you are not very good with your budgeting or the budgets are tight, in other words. And then the third thing is the technology changes, because what happened, building is designed for a particular type of activity, and then this same building they start like doing for some other businesses, then that's going to be a problem. Down this presentation we can also give you some good example on that. Then the HVAC contamination. HVAC is a word typically refer heating, ventilation, and air conditioning. So that itself is a component which generate lot of contaminant, because while we are conditioning the air, we remove lot of unwanted debris and unwanted um, entities from the air. So if we do not manage it in a right way, then that discarded material may be problematic and maybe penetrate through the livable environment. And the occupant concerned, because some of them are get the media news or somewhere, if they are convinced that that this kind of sickness is happening through the mold, then they basically think no matter what, I am sick with the mold. So that is the another consequences, that is like psychological implication, I would say. So not all the time it is a true statement, but many a time we all start thinking that if some epidemic or some episode of mold is being published, and then we start directly correlating with ourselves or come on the individual level, then that catalyze the whole issue about exploring the mold. So these are the few I said you the focal point by which we can say that the whole mold origins on the worriness about mold come in human society. Now, Ronnie, it is also important to understand that how mold grow. That is a very, very important, information. I want to explain our viewers, our audience, that the mold growth is like very common if you meet four basic parameters. What are those? The very first parameter, I would say, moisture. You have to have moisture. Then the second one, that you have to have adequate temperature. When I say adequate temperature, meaning that it is about like 65 to 95 degree Fahrenheit. Once you met with these two conditions, then you have to have mold spore, which you have no scarcity in the nature, because as I said earlier, that mold is ubiquitous in nature. So lot of spores are in air already, so they are going to incubate themselves once they settle, and that's how the proliferation start. And of course you have to have the organic food material, or the food material, in other words, is the material which mold typically decompose, because mold has a very important or significant role in the ecological cycle, that they are decomposers. So the complex organic material getting simplified minerals and elements is going to be deteriorated like that. So if you have these four parameters, then I will be not surprised, and there may be some issue regarding the mold growth, that not always going to be bad or good.
Okay. So now we're going to go ahead and launch our second question to make sure everyone's still with us. Why does indoor air quality matter? Is it energy conservation, health and hygiene related issues, both A and B, or none of the above? Since y'all seem to be pretty quick about answering this, go ahead and give you another 20 seconds or so. Okay, I'm going to go ahead and close the poll now. Let's see how everybody did. And it looks like 66% of you got it correct. Not as high as the first one, but we still have some of you with us. It is both A and B. It's important because of the energy conservation, and it does affect the health and hygiene related issues, both of the building itself and of the occupants within the building.
That's very good. Looks like our audience is getting most. We are trying to get the point across. But like Ronnie, you asked me that, like, I said you the factor for growing the mold. But at the same time, it is very important that where is the most suitable place in a building where you can see the mold growth. These few pictures are elaborating that finding, or elaborating that statement that the mold can grow in hot and humid place. As you can see, mostly in the energy tight building, you have a duct work system. The duct work system is going to be like an incubator of mold if it is not being properly managed. The duct works are like a baby diaper. It should not be like dirty, it should not be like wet, and it should not be smelly. So if you avoid these three situations, then perhaps you are minimizing the risk of mold. But as you can see in these pictures, that if there is moisture, if there is enough organic material, which is always going to be there, then I will be not surprised there will be mold, and obviously that will give you the dirtiest smell, that is sometimes mustiest smell people refer at. Same thing here, you can see in the different situation or different positioning within the duct, the mold start growing, and it grow in copious amount, and it send downstream air lot of viable or non-viable mold spore or more particulates. Some other places where you can get the mold is basically water intrusion, or like if it is a post flood scenario. These two pictures which I am showing you, that give you an idea that how the receding water has make the building material richer in the moisture, and that moisture has supported the mold growth because there is plenty of organic material there. And as I said, the spores are in the air, and then no wonder that if it is a hot and humid, the mold start proliferating. Is that something makes sense, Ronnie?
Yeah, makes perfect sense to me.
The other thing I want to give some more information. People often ask me that what are the common mold in the indoor environment. Well, if I start counting one by one, then there is several mold here in the indoor environment. Working with this laboratory in past 2 decades, I think more than 150 different type of mold I have reported on various samples. But few of them which is very frequently encountered in indoor environment are Alternaria, Aspergillus, Chaetomium, Cladosporium. I'm giving you a little bit of pictorial view so that you can remember that how mold looks, because obviously you cannot see this mold with your naked eyes. Then Carvularia, Penicillium, Stachybotrys. This Stachybotrys is often referred as a black mold. So you can see their spores are like coffee bean sepid. They produce rather coffee bean sepid spore. Now, I was telling you that mold can be also in air. This particular slide is showing you that various kind of a spore, and I don't want to make anybody scared, but this is a real slide, real world example that the air was having all those kind of a spore, which I have for convenience mark them with number 1, 2, so that you can recognize those are the mold spore which we have identified on that particular slide. Then other stuff which can also be trapped with mold from air is given on the right hand slide, which basically telling you that a number of other debris including the human skin cell, black particle, etc. help proliferate mold, because that becomes like sometimes the food material for get going the mold in the air. So it is another question that whether surface borne is the result of airborne or airborne is the result of surface borne. So now, what would be the high risk area? The high risk area for mold growth is basically identified as dark, hot, and humid areas. Construction zone where you have lot of nuisance dust, because that dust can carry like lot of organic material. Obviously, moisture is needed, and then temperature. Summer cottage, because summer cottage, like many a time the water is being sprayed, and people try to make it like cool or cool down the scenario, so they make moisture going in that, so that is also a very high risk area. Farmland or the farm itself is like some of the buildings are not properly like functioning, so that might be also a very good place for the mold growth. The florist don't it's not against the florist, but in the florist the situation is very suitable for the mold growth, because they have like various twigs and other part and pieces of the plant, including flower, that might carry some mold, and if it is being not processed correctly, that can be airborne or surface borne. It can go either way. Greenhouse is the another possible potent area for the mold growth. Antique shop, because like if you have various article touched by or pass through the various like hands, then if we contribute like mold or some contribution happen through the contaminated hand or dirty hands, then that is going to be there, and that can be very well like inert on that piece of antique, but over the course when the favorable condition come, that can start like growing. The saunas are the another potential and risk area. Perhaps you can understand that there is a lot of water activity in the saunas, so those are some high risk potential area for the mold growth.
Okay. And yeah, and I just want to kind of bring up another example. These seem to be like a lot of very more permanent or more consistent hot and humid areas. But even sometimes, some temporary things can happen. I can't count, how many times we've had a customer send us a sample. They've had mold all over it, and they say, oh, where did it come from? Why is it the case that we have so much mold? And I ask them, okay, where'd you take the sample from? They tell us that it was in their basement and they had a leak a year prior or a few months prior. And they're surprised to hear that. But even something such as a temporary flood or a leak can even lead to this, very prolific mold growth. Because once again, following your logic for the mold growth factors of hot, humid, you have a food source or anything like that.
Exactly. So how the mold is like affecting our health and hygiene? Well, there are four broader category which I think is very important. The mold is going to give you a lot of byproduct, as mold itself is a decomposer. So when the mold is growing on a substrate or a substratum, then it is going to decompose. Some of the product after decomposing that complex material is going to be ecological friendly or friendly, and some of them are not going to be that friendly. For example, if the byproduct is methane, then or acetone, then that is not going to be very human friendly, so you are going to have some effect out of it. The second one is the exposure. The exposure of mold can happen with three possible way. First is inhalation. If you inhale some kind of mold spore, pod pieces, etc., and you are susceptible to it, then you are going to sensitize by that. The other one is the contact. Like if you touch the high surface area or a high touch area, and if it is contaminated with the mold, then obviously you get also compromise with your health and hygiene due to contact of mold. Then the third one is the ingestion. If you eat some contaminated food with mold, then obviously sometime the food poisoning or the mold infection is going to happen if you are susceptible. So that's why I put the third point, susceptibility, because not every individual has the same immune response. So the people who is having weaker immune response are more susceptible to acquire infection in comparison to otherwise. Then the fourth point is the toxicity. If The toxicity basically depend that the nature of mold and the chemical or the biochemical which is encompassing the whole structure itself. Some mold are capable of producing mycotoxins, so they are more potent and they are more toxic in nature than otherwise. So these are like some basic point we need to understand when we start like thinking about the mold and its health and hygiene implications.
And you talk about, you just mentioned how mold can interact with us. What are some of the effects of that interaction for the inhabitants?
Well, so far the mold effect is concerned on the human health, then possibly there is another 3 point I would like to bring forward. The first one is the allergenic reaction. Allergenic reaction is capable of producing like allergenic response such as wheezing, coughing, itching of like skin, watery eye, nasal discharge, etc. These are, or sometimes like mild fever. These are like some very non-specific symptom which is being like responded upon exposure to the mold spore. The other one is called as pathogenic, in which the organism go and establish in your body system, and then you show certain symptom of infection, that is like pathogenic aspect or pathogenic effect of mold. And the third one is the toxic mold, when you have like getting exposed to those mold either through inhalation or contact or ingestion, you are going to have certain symptom. One example I want to give you with the toxic mold such as Stachybotrys in the Ohio Valley. This is being reported that a lot of kids get like sick, and upon exploration, the medical investigation lead that those kids were exposed to the Stachybotrys, and actually it start like infecting their nervous system, meaning that their brain, and that's how they get sick, and some of them actually died.
Okay, so we'll go ahead and ask our third poll question now. And the question is, where does mold exist? Is it in the air only? Is it surface only? Or is it both air and surfaces? Or is it none of the above? Go ahead and give everyone about another 20 seconds or so. All right, looks like most of the votes are in, so I'll go ahead and close the poll now. And it looks like everybody did incredible on this one. It is indeed both air and surfaces where mold can exist. And although we don't have it listed here, we did also mention that it can be found in liquids or fluids as well. All right, Dr. Sahay, so we have a basic foundation of mold, what it can do, about the indoor environment, inhabitants. Can you go through some of the parts of investigating mold in an indoor environment?
Very right, because once we have that background information like any other investigation, I think 5W's rule is also very good while we are investigating the mold. What are those 5W's? Obviously, who, what, when, why, and where. So we have to find the question that like who is complaining, what the complaint is about, when the complaint was filed, why he is complaining, and where the complaint is. So if you go in that sequence and start like using the process of elimination, you will start finding yourself in a position where you start narrowing down the actual target. In this case, that is the mold growth. So that makes sense, Ronnie?
Yes, very clear.
So how the investigation start, what are the factor which basically incorporated in your thinking so that it lead you to a very good result. The very first thing, there should be motivation. Obviously you're not going to tell me that okay, money is the motivation. Yeah, sometimes maybe, but some people is motivated to find out that what is bothering them. So the motivation is number one, that what like start the process. Then obviously history or complaint is the second aspect of any investigation, because if there is no complaint or there is no history file on that, then there is no investigation it, as possibly you can think for any other scenario. Then the third step of investigation is walkthrough or evaluation. Like I said you that, like, it's a process of elimination. Just start, like, selecting the obvious, and you will be start, like, drawing yourself with the target. The fourth point, which is a very important point from the laboratory point of view, that the samples should be collected, not only collected, but samples should be adequate, samples should be well defined, and samples should be preserved, so that the laboratory can start, like, investigating that. So the fifth point is the laboratory testing. Unless and until you perform the testing, you will have no idea that what kind of mold is proliferating, or what kind of mold is infesting in that particular substratum or the material. Once you explore that, then you are going to generate certain result. So that result is going to be a very key factor in this whole proposition, that it, once the interpretation of result or the finding is done in, like, adequate manner, then it lead you, or it give you the point of management. If you know something, you can manage it. Without knowing, the management is out of question. So if you know it, you manage it. So implementation of the result is going to be the true implementation, and it is going to help in a big way to solve this problem, if any, or existing problem. And then obviously many a time, all of a sudden the emergency happen, because if somebody start, like, showing symptom rather quick, then you don't have to time for, like, the doing lot of investigation. In that case, I would say that, like, collect few sample, look around, see around kind of approach, and then you can send those sample and ask them to do the testing as quickly as possible, so that can be responded on time. So the motivation is basically the proactive approach or maybe reactive approach, because a lot of motivation is just, like, initiate yourself to be proactive, so that you don't deal with this kind of situation. Some motivation just come after the reactive response. Like if somebody start complaining, then you kind of get motivated, really it is making him sick. So that is a reactive approach. And then of course, like, emergencies are there always. Like if you see, look around and people are complaining, then it's sort of emergency situation, so you can take on that. Now the history or complaint, basically what you have to do about that, you jot down the any problem being forward for the health and hygiene issue. Occupant complaint, you should pay proper attention. Just do not start thinking that the person who is complaining is just, like, not understanding. But many a time, or majority of time, it has been noticed that a lot of people has no idea about mold, but they are giving you a specific or non-specific symptom that can be related with mold. So the occupant complaints should be given with a due consideration. The persistency or the persistent versus temporary, that the problem, if an occupant is coming, whether it is a persistent problem or it is a temporary problem. Then the moisture intrusion, like you have to pay attention, because moisture is reason for mold, no doubt about it. Everybody agrees on that. So you must be investigating the possible area where the moisture migration happen or moisture intrusion happen. New furnishing is another thing. Like if you are trying to build up or adding some building with the another, like, adjoining area, and if, like, the somehow the other the building envelope and other thing failure, then there is a moisture migration, then obviously new furnishing is also going to show you some mold growth. Same this program, this what logic I said you is apply in the renovation and construction. And obviously the HVAC work, if it is not done right, that itself a big warehouse to generate the mold. So what you do in the walkthrough? The walkthrough is inspection is at a glance with due diligence. When I say due diligence, meaning look, see, smell. So the visual assessment and then unusual odor detection. So if you have some unusual odor which is like not explainable, then you look and collect the sample so that you can test it later to find out that what's going on in that particular situation. The next one in that one is the determination of the causal factor, perceived versus the physical symptom. Sometimes people think that they are like sick, and they have no reason to believe that I'm not sick due to mold, because they are going to perceive, they are keep telling you persistently that like, oh, I'm sick due to the mold exposure. That is one facet or one coin of the one side of the coin. Whereas there is other people where you can definitely see some physical sign, like as I was telling you, runny nose, itchy eye, etc. That's a physical symptom. Then you have to also see the frequency versus timing. It is happening like, in particular time of the day, or it is throughout the day and night. Individual versus group. An individual is complaining or the whole group is complaining. Maybe in office one person complain, one lady do not want to work, she can complain, or one gentleman who do not want to work on that day, he's going to complain. So we have to see that whether it is individual or is a group. And then the source causation relationship is a key factor which help in Big Bay during the investigation to find out the mold and the mold growth and associated factors. Then the indoor versus outdoor, because mold is ubiquitous in nature, not necessary that the mold is going to grow in indoor. It might be very easy infiltrate from outside. So whether it can be outside mold or inside mold, it doesn't matter, because if you have mold, then you have to respond. It's a natural versus induced. Sometimes mold is a natural phenomena, it's occurring naturally, you do not have a lot of control. But sometimes mold is induced, because if somebody is walking from a mold contaminated area without taking adequate precaution, then they are going to introduce the mold in non-contaminated area, or possibly where the mold is not growing. So that is the third point of the investigation, which is the determination of the causal factor.
And before we go continue, next with the samples, I'm going to go ahead and launch the next question for everybody. And it should be up on your screen. Which is a supporting factor of mold growth? Is it moisture, temperature and humidity, mold spores, or all of the above? Go ahead and give everyone about 20 more seconds to get their answers in. Okay, I think we got most of the answers in now, so I'm going to go ahead and close the poll. And we'll see. Looks like everybody's still paying attention with us. 100% of you got it correct. It is all of the above. Glad we got that point across about mold and its intrusion in the indoor environment. So now we'll go ahead and continue. Dr. Sahay, if you'll go ahead and speak about samples.
Exactly, sample proposition is a very big part of the investigation. So a lot of people ask me that what kind of sample we are going to collect from the environment. That's a very good question, because environmental sample come from three major category, which you can see right hand side listed on this slide: air, surface, liquid, water, or we can call fluid. So these sample or these category must be authenticated, meaning that if you are collecting this sample on a particular site of the building or in a particular position in the building, you must authenticate that sample was collected from there, because later it is going to help you to identify the source, which I have jotted down as a point number 2. And then the result when we test these samples is going to tell you the enormity of the or enormity determination, meaning how bad or good the situation is, what kind of mold we are recording, or what kind of mold we are identifying, and what is their quantum. So that's very important, I guess, aspect of the sample, and especially the environmental sample, because environmental sample are a little bit different than clinical sample. Typically in clinical sample we go after one target, but in environmental sample we do not have a target. We want to investigate or evaluate the sample in for their totality. that whether it is mold contaminated or not. If it is contaminated, what kind of mold is there? If there is a mold, how much mold is there? So let's start with the sample type. Very first type of sample come in my head from that matrix is called as bulk sample. Bulk sample is nothing but it's a portion of environmental material collected with an intent to test it for a specific interest. For an example, that sample could be like a gram of dust, some building material, wooden plank, and etc., some pieces of furnishing or furniture, etc., that can serve as a bulk material or bulk sample. Now, the other very common sample is called as swab or wipe sample. That is being collected from surface to assess the surfacial constituents collected by swabbing or wiping a surface with intent to test again with a specific interest. That is a swab or wipe sample. The next one is the next one is going to the air sample, which basically volume of air is drawn by employing various kind of equipment with right now there is a lot of air samplers are known, and collectively that technique is called as a spore trapping technique. So you can collect the sample using the spore trap technique if your goal is to understand the overall scenario of mold. But if your goal is to understand the viable mold or the mold which is actively growing or grow on the culture media, then you can use an impactor. Again, the principle is same. You suck the air, or just air is collected over a microbiological plate so that you can see what kind of mold is going to grow in that. The other method or sampling method is called the impingement method, in which again you draw the air on a liquid and that kind of growing in that liquid, and that's how you find out whether there is a mold contamination or not. And obviously the filtration is the another method by which you draw the air and collect dust and debris in that air, and then later you can either do the microscopy or you can do the bulk culturing method, meaning you start culturing it to see whether any microbes or the mold is growing on that or not. So some other method, which is a very common method, is called as gravitational settling method. It's a very simple method. You can have a media plate. When I use the word media plate, it's a microbiological media, and then you open a place or a desired location for some time, and then you close the media and then transport it to laboratory and let it grow for a few days, and you can identify the mold. That is also a very efficient, I would say, quick screening method, but is not very valuable in terms of quantitation and uh, in terms of exposure limit or determining what kind of mold, because there may be some issues. You do not know the volume of the air in this. You typically do not know the how long mold takes to grow and what kind of temperature is there, because some mold can grow on room temperature, so they are going to outbeat the other mold. When you put in the incubator, then they are going to grow. So this method is not that effective. The thermal precipitation, meaning you heat something, maybe heat a metal plate and then you let it cool down, then you will be having some kind of precipitation. You can test those under microscope, or you can also culture them. The condensation trap method, the another method. And then the modern time, a lot of people are trying to study the mold and other microbes of the environment using the electrostatic precipitation method. Those samplers are getting popular, so that you can also collect by using the principle of electrostatic, and you can just investigate by doing either microscopy or culture. Now, at this point, I would like to tell you that what is type of analysis laboratory provide. Well, the very first type of analysis is called as non-culture technique, in which we do not culture anything. Our intent is not to grow something out of the submitted material. Rather, what we do, we enhance those material by using microscope or added eye, so that we can see what is going on that particular area. That microscopy can be bright field, polarized, electron, etc. So you can see by enlarging the viewing area so that you can find out that whether mold is growing on that particular material or not. There is another non-culture technique which is, I would say, very efficient. It's called as enzyme linked immunosorbent assay. In other words, ELISA. In this method, you collect the material, you extract the material, and you run it against the standard, and that way you can find out what kind of mold is growing. The other one is gas chromatography, mass spectroscopy, that basically investigate the chemical, and based on the chemical structure or chemical combination, you can guess, like, what kind of mold that might be. And other method of doing the chemical testing or MBO testing is high performance liquid chromatography, that is also listed under the non-culture technique. The next one is the culture technique, in which we use microbiological media that we have already potentially identified that what kind of formulation or what kind of food microbes like, especially mold likes. So we use those kind of media, such as you might heard laboratories saying that it was grown on MEA, TSA, or, like, several other media, JPEG stocks media. So there are several different type of media. MEA, which is a very common media, that stand for malt agar extract. So that, like, you can ask the laboratory, they can give you more detail on that. There are certain tissue culture technique by which we can also grow certain mold. The microsporum come in my head. This is like a nail fungus, so it can grow on the live tissue or dead tissue, and that way you can just have the growth of that particular mold for their particular identification. Other technique is the molecular diagnostics we use for mold identification. One of them is called carbon source utilization, or in other words, we also call it fingerprinting. Because if certain mold is, like, doing certain type of reaction, when we let it allow, or let the mold being allowed to grow or react with the source of carbon, then they give you a printing, a pattern. So computing that pattern, you can identify that type of mold. The other one is called as polymerase chain reaction, in which you can take like a part of DNA, or extract the part of the DNA of the mold, and you proliferate it, and you like amplify it, and that way you can identify that particular mold. So that is polymerase chain reaction. Now, after the investigation, obviously you get a result. So the result interpretation, I guess, is one of the important part of the investigation. So what, or how you interpret the result? The very first information you try to derive from those results, that whether it is a quantitative or qualitative. So qualitative versus quantitative is going to help you to understand that what kind of mold you are dealing with, and what are the quantum of that mold that laboratory finding or the result is going to tell you. The living versus non-living, whether the mold is an actively growing phase or non-actively growing phase. Biological versus chemical, like if the mold is giving you the biological factor mold versus, like, it is generating some MVOC or mycotoxin, etc. So that gives you an idea. The technique itself narrate a lot of things the laboratory uses to test the mold, such as microscopic versus visible. So if the microscopic technique is being used, then you are seeing for real, like, the spore, and also your identification is a little bit more clear than the visible, because visibly you can see the mold, but it is very hard to tell just by looking at the mold colony that what kind of mold is growing. The allergenic versus pathogenic, again, the proper identification can lead you that this mold is like listed as an allergenic or allergen versus a pathogenic. The investigation can also lead you to draw the conclusion whether the mold is actively growing or it is going to cause some passive impact. And of course, a lot of laboratory has their own way of interpreting, which they typically lot of laboratories share with them. Our laboratory ED lab also share what is normal versus abnormal based on the sample. We have analyzed over 400,000 sample till date. So based on that, we have extracted some good statistics to share with our customer that whether normal versus abnormal. But we at this time do not have universally accepted guideline. So I can just supply to the end user and they can use it. So it is pretty much due diligence and some supportive factor or supporting data which we share with our customer to determine whether they are dealing with normal versus the abnormal condition. So that was pretty much about the laboratory interpretation, Ronnie. The other thing I want to tell, if you are investigating the surface, many a time our viewer asks what is normal and what is abnormal. That's a point blank question. Don't you get that kind of question all day in the laboratory?
We do, yes.
And for answering that question, I incorporated this particular slide to just give you an idea that what kind of condition you are dealing with if your lab result come positive for mold, and what should be the remediation or what should be the action you should be taking. For an example, we call condition 1, which is normal. If you have 0 to 50 count of mold spore or mold particulate growing per centimeter square, then basically that most probably you are dealing with the settled mold spore condition, and we list as in condition 1. If you have more than 51 to 175 count per centimeter square, perhaps you are dealing with the condition number 2, which we listed as action needed, and it is basically resultant of the settling mold spore with some growth. But when action is warrant, if the concentration of mold is 175 count per centimeter, and that is actively growing mold, and we listed those things as a condition 3. Now, the air mold or fungal contamination, if we are using the null culture method, if the spore concentration is 1000 count per cubic meter volume of the air, that's sort of low range. But if it is getting or topping more than 2000 using the doubling effect, that is in a high zone and the action is warrant or action is necessary. If you are doing the investigation using the culture method, then the concentration supposed to be 350 colony or less. If you are topping over 700 colony or something, action must be taken to prevent the mold growth. Next one, please. The management wise, we need to address the findings, whether we are dealing with the allergenic or non-allergenic, pathogenic versus non-pathogenic, viable versus non-viable, and active versus settled. So the action for different scenario is different, because if it is an allergenic mold, then we need to find the way that how we can minimize the growth. The only way to minimize the growth, or important way to minimize the growth, is minimizing the moisture intrusion in the building. So more moisture migrated, more mold growth. That is the logic here. The next one is basically you need to have an action plan implemented for managing the mold growth and the mold situation, that include but not limited to the corrective action. If you find the mold, you need to remediate it. You need to stack the mold for the livable environment, and then the remediation is the process by which we can take out the mold, we can correct the situation, and we can bring the normal. Other action plans should be preventive action. Preventive action is saying that you don't lead in that kind of problem. You can just restrict yourself with certain parameters, such as you have the baseline information that what is growing in your building, so you can just limit the moisture migration. You can do the housekeeping practice. You can just not let the dust settle. So these are the practices by which you can just minimize the growth of mold. Try to maintain the temperature optimal level, most probably 70 2°F to 76°F is a good temperature range. You can also control the moisture by controlling the relative humidity. That should be somewhere between 30% to 60% is a good range to be maintained, so that you can minimize the mold growth. The routine maintenance is the key of management. So a routine management is basically look, see, and act. So if look, see, and act is in place, then you will be minimizing the mold growth. So in summary, the common indoor contaminant is also mold. So mold is not like something else, but it is something common contaminant on the indoor environment. It is very important from health and hygiene point of view, like as I said earlier, that this is going to create some problem, maybe a hygienic, pathogenic, and toxigenic effect. Identification and quantification of the mold is basically help in improving the indoor environment quality as a whole, and management of indoor environmental quality or indoor air quality is not very difficult, not very time cumbersome, not very technical. Again, simple approach, look, see, act is beneficial, so that we can just prevent to getting mold and face all the consequences. And last but not least, I think more pixels always better, better, better for resolution. More information you gather, more good management you will be capable of. So I think this should give us good information to our viewers.
Yeah, and that is the conclusion of this webinar, but we do have our very last question, and I know we're just about out of time here, so we'll go ahead and make this last one very quick. At what level is action needed in case of surface mold contamination? Is it 0 to 50 counts, 51 to 175 counts, greater than 175 counts, or none of the above? This question is a little bit more tricky, so we'll see how everyone does on this. And I think we've got about most of who's going to vote in here, so I'm going to go ahead and close it now. And it was very close, but it was the 51 to 175 counts. That greater than 175 was considered action required rather than action needed. So thank you all for joining our webinar on mold in the indoor environment.
And again, thank you very much from the ED lab. This is Dr. Sahay, and we will be a standby for any other question. You can contact us via phone, email, or whatever way you want to. Even you can write us the letter. If you are not computer savvy, we can definitely respond to your question. And we love to spread the information on mold, and we are not shy to share our experience with the mold in especially indoor environment or building. Again, thank you very much for joining us.